Hi everyone, my name is Huiyun. I'm an ORISE postdoc working at EPA RTP campus in North Carolina. Today I'm going to present you one of my PhD program's work. It's about microbial source identification using a concentration-discharge diagram. It's an application in a central Michigan River watershed. Microbial contamination is one of the most pressing environmental concerns. Therefore, development of watershed management plans for TMDL sites is needed and urgent, especially in the Great Lakes area. There are many sources of microbial contamination. They may this is from pet, wildlife, livestock, and they may from discharges from wastewater treatment plants, surface runoffs, combined sewage, combined sewer overflows. The hypothesis of this study is that E. coli and bovine bacteroides have different transportation pathway in water systems. And the objective of this study is to first investigate the potential origin of microbial contamination in water systems, whether it's from surface water, soil water, or groundwater. And the second one is to apply the concentration-discharge hysteresis loop to understand the origin and the transport of microbial pollutants. In this watershed, we have a mixed land use areas, and in the sampling site is on MSU campus because the Red Cedar River flows across the campus. And the upstream has a lot of farmland, and there are 3 wastewater treatment plants discharge into the river, and there's a K4. There are 3 K4s in this watershed, the concentrated animal operation, and then they may be significant sources of the microbial levels in the sampling site. Therefore, we first selected the site because this is defined as a TMDL site by Michigan DEQ because of its elevated E. coli concentrations. We choose E. coli and bovine bacteroides as the microbial contamination indicators, and then we generate the CQ hysteresis loop based on the concentration level and the discharge from the river, the river flow rate. Then we get the information from the loop's direction, amplitude, and slopes to understand the timing and the sources of the water component that carries the microbial contamination into the river system. We choose E. coli as an indicator because this is one of the most populate microbial indicator in many research. And we also choose bovine-associated bacteroides to identify the fecal contamination from bovine or cows. This is a 16S RNA gene from bacteroides. It's like a name tag, and we can get the host information from this gene numbers. We take graph samples from April 7th to June 17th in 2013, and the sampling day sampling period covers 71 days. E. coli was measured by culture method from the coli 18 kit. Bacteroides was measured by qPCR. First, we concentrate the stream water from 500 milliliter to 2 milliliter by filtrating the samples through 0.45 micrometer membrane filters, and then elute and recover the filters from some buffer and get down to 2 milliliter. we get DNA extracted from the concentrated samples using Microbial compact system. This is an automatic machine, and we use this corresponding kit. The qPCR was carried by the light cycler Roche machine with this probe master kit. E. coli was detected 100% in the sampling events, and out of 71 samples, 22 samples were measured exceeded the water quality standard, which is 300 mpm per 100 milliliter, and that's 31% of the sample. And you can see that some of the E. coli peaks occurs before the hydrograph peak, and some of them occurs after, and this effect is called the hysteresis effect. And based on this pattern, we can generate the CQ hysteresis loop. The discharge was measured by the USDA scale on the river. Bovine-bacteroides was measured 100% as well. The highest concentration of Bovine-bacteroides was on June 6, 2013, and the timing is similar to the highest E. coli peak. And you can see that this peak of BoBac occurs a little bit before the hydro peak, and then some of them occurred around the same time, and also this would help us to generate the hysteresis loop. In order to understand the bacteria better, we also detected nitrate in the system. Nitrate was the highest concentration of nitrate was on April 13th, 2013, and this might be because of the snow melt and flushed in a lot of nitrate was flushed into the river from the snow deposit. And we can also see that this peak occurs before the second peak of hydrograph, and a little bit delayed from the first peak. In order to generate the concentration flow, concentration discharge hysteresis group, we have to divide the sampling events into different hydrologic events based on the shape and the peaks of the hydrograph. Basically, in this study, we divided it into 3 events. The first one is from April 7th to 17th. The second one is from April 18th to 30th. The third one is from May 25th to June 16th. And the bar is the precipitation. We didn't include May 1st to May 24th because the microbial contaminations level are very constant in this period. I'd like to introduce you the fundamental concepts of CQ diagrams, which is the different water component that brings or carries the contaminant solutes to streams. There are 5 pathways of water coming to the stream. First is direct precipitation. The second one is surface runoff. The third one is interflow, and the fourth one is bank flow or baseflow. The fifth one is the deep baseflow. And if we put this component into a hydrograph, we would generate a hydrograph like this. The deep flow is constantly flowing into the river and is the base of a hydrograph. And then there is the bank flow or base flow, and then this is the interflow, and the the top is the surface runoff, because the direct precipitation input is very minor into the system, so we ignored it in this graph. Then we simplified these 5 components into 3 components, which comes to surface event water corresponding to surface runoff, soil groundwater corresponding to interflow, and groundwater corresponding to bank flow and deep baseflow. The CQ diagram would give us this information for direction, amplitude, and slope. When we generate the CQ diagram, we plot the concentration levels on the y-axis and discharge on the x-axis. The slope of the diagram would tell us whether it's a flushing or diluting event. If it's greater than 0, it is a flushing event, resulting in increasing concentration in the water of contamination. And if the slope is 0, it results a diluting effect, meaning the concentration levels will be decreasing in the event. And the amplitude of the slope will tell us how long the intervals between the peaks of concentration to the peak of the hydrograph. And I will explain it in more details in the real examples. So there are 6 scenarios of the CQ diagram based on the direction of the diagram and the slope of the diagram. And these are the dominant sources, the order of the sources of the 3 water components. For example, if it's a positive slope in a clockwise direction, it means the surface water is the dominant source, and the second one is soil water, and then groundwater. And we can tell these things for other scenarios. For example, if it's anti-clockwise direction and the slope is positive, then soil water is the dominant source, and then it's the surface water and groundwater. Usually, if the dominant source is from soil water or groundwater, it means that the source is coming from a far distant location. Whereas if it's a surface water, it comes from a very nearby location of the sampling site. We plotted our CQ diagram here, and we can see, for example, bovine bacteroides in event A has a negative slope and anti-clockwise direction. So this would be something like this A3 diagram. And we can tell that groundwater is the dominant source, and soil water is the second, surface water is the last. And for example, another example is for this one, E. coli. This is a positive slope, and it's the anti-clockwise direction. So we can go back to the diagram examples and find out that this is a positive slope and the anti-clockwise direction. So soil water is the dominant source, and then surface water is the second, groundwater is the last. Therefore, by comparing the examples to our plots, we find out that in the first event in early spring, E. coli has surface water as the dominant source, but bovine bacteroides, the dominant source is the groundwater, and the nitrate is from the soil water. And then in the later spring, soil water becomes the dominant source of these 3 indexes, But in early summer, when people start to apply bovine manures, the soil water, especially surface water, become the dominant source of both E. coli and bovine bacteroides, so as nitrates. So if we look into the details, we can see that first the environment in the early spring, there's some deposit of E. coli on the ground, and then it is flushed into the river. So there's no more contamination from the surface. Soil water become the dominant source, and then after the manure application, surface water become again the dominant source. Whereas in bovine bacteroides, there was not many contamination on the surface, so groundwater was the dominant source, and then soil water and then surface water become the dominant source. This might become because bovine bacteroides is anaerobic bacteria, and it doesn't amplify by itself in the environment. So it represents the fresh fecal contamination from bovine, and whenever there was signals, it represents the fresh contamination levels. Therefore, we can see that E. coli and bovine bacteroides share different transportation pathways in early spring, but when it comes to summer, they share similar transportation pathways. Therefore, by generating the concentration flow discharge diagram, we can understand whether it is a flushing event or the diluting event, and we can understand what is the dominant source of the contamination that carries the contaminants into the river systems. This information can be useful to help us make water management plans and to make sure that we can capture the flushing event and make sure that the water contamination is below the standard for the study site. I would like to acknowledge our collaborators, which includes my PhD advisor, Dr. Irene Zagraraki, and my committee members, Dr. Boyce and Dr. Long, my lab mates, Dr. Amira On, Ao Chen, Fang Li, and Ensang. This is our teamwork, and we took samples and work our own parts on this project. Please feel free to ask your questions. I'm happy to answer your questions. Thank you.